Thank you for the opportunity to speak with you today about the Alzheimer's genome. I think everybody in the room probably knows that the causes of death for age-related diseases are decreasing for everything except Alzheimer's and Parkinson's disease. There are an estimated 6.5 million Americans above the age of 65, at the age of 65 or above, with the disease. And by 2050, it's likely that we'll be spending $200 billion a year. In 2012, Congress passed the National Alzheimer's Project Act with main goals to prevent the onset of Alzheimer's disease and to develop effective treatments for the disease by 2025. Simultaneously with the launch of NAPA, the ADSP was launched in 2012, and we do reporting annually in 3 milestone categories: epidemiology, molecular pathogenesis, diagnosis, and biomarkers. The project started its sequencing in 2014, and we had small numbers of whole genomes at that time because we didn't really know how to analyze them, and they were extremely expensive. We moved then more boldly into more whole genomes, recognizing that there wasn't very much going on in the exome, and then up to 37,000 whole genomes beginning in 2017. And now this summer, we've launched an initiative that I'm going to talk about for most of the rest of the discussion, to bring in 150,000 whole genes with subjects with diverse ancestry and as much omics as we can possibly get, along with the phenotypic data. The goals for this part of the project are the same as the rest of the project: to identify new genes associated with the disease, to identify genetic variation contributing to increased risk for protection against the disease, to find out why individuals who have known risk factors can escape from the disease, to identify potential avenues for therapeutic approaches and prevention of the disease, to look carefully into multiple ancestry categories for their genetic architecture, and to share the data rapidly with the community. The ADSP enjoys robust support from the National Institute on Aging from several places: 37 Alzheimer's disease research centers. NAC provides the clinical data. NECRAD is robust in terms of providing support for banking of DNA, plating of DNA, and our cell repository, tissue repository. We have a large family-based study that Richard Mayu runs, one single large-scale sequencing center, one single genome center for Alzheimer's disease, doing all of the quality control checking and harmonization for the project. And we have a database, one database, for broad sharing with the research community, providing cloud-based data access, and it is VISMA compliant and equivalent to dbGaP. And we've added this past year a large cooperative agreement for phenotypic harmonization to harmonize all of the phenotypic data for the ADSP. So we have now at least 350 investigators, 5 large consortia, individual grants and cooperative agreements. These are supported by NIA and an external advisory board that is a blue ribbon global panel that gives us support when we hit speed bumps and lets us know when we're doing things right. So all of the consortia talk to all of the other consortia, and we have a number of work groups. We have an analysis coordination group that's has endeavors related to policy and publication, and an analysis work group that does problem solving, and now is also featuring monthly work groups to feature the important outcomes that the ADSP is generating. And with this, we have domain experts in 14 areas, 14 different types of data that we have for the ADSP, 2 domain experts in every area. And these are this team is coordinating all of the work that is being done. So this is a famous diagram by Andy Sakin that I want to say pretty much holds from what was thought from in 2015. But I want to change the perspective a little bit and note that we are now gearing our gene discovery towards driving us towards defining endophenotypes. So we don't have many common variants. If you look here in the discovery component, we have a lot of rare variants. Almost everything is rare and intergenic. This is not a monogenic disorder, and it is a family-based disorder. The functional genomics criteria have entered over the past year and a half into our pathway here. Understanding the mechanism requiring development of additional research resources such as stem cells and functional assessment and gene editing are underway with a functional genomics effort. The therapeutic hypotheses have been pretty well established. I think there may be a few new ways of looking at the data, but largely being pathway driven now. One thing I want to point out is I want to move this biomarker development over here because the biomarkers are really pointing us towards the genes and the endophenotypes. So this would be one change that I would make in Andy's diagram. So why are we studying the genes? I think probably everybody knows we have to understand the biological pathways. And with that, we have to understand the mechanisms of disease. We're in a place now where we can begin to do prediction models with polygenic risk scores for things such as age onset or severity of disease. Again, the biomarkers are really important for what we're going to be doing, pointing us more and more to potential therapeutic pathways. And I am happy to say that we have, evolving from the ADSP work, we have probably at least 3 clinical trials either underway or just about at the stage where they're ready to launch from ADSP work exclusively gene-driven. So the history of this disease for the genetics is quite long. It started late in the 1980s with familial Alzheimer's disease genes being identified, presenilin-1, presenilin-2, APP, and also APOE4 for the later onset Alzheimer's disease. But first of all, in this area, everybody thought the disease was mostly going to be sporadic, but it isn't. It's mostly genetic. And we were in a gene desert between the mid-1990s and 2010 or so. And I stepped into the portfolio right around here. So there was a long, hard time in the beginning part of what the investigators who are now in the portfolio were doing, where things were really pretty difficult to understand because we weren't coming up with any genes. Then GWAS happened and things began to move along pretty swiftly after that. We began to see pathways in these loci, and now whole genome sequence data has been added, and this is robustly supporting more gene discovery. So before 2021, what I was seeing was predominantly APP processing in the presenilins from early on as a pathway, but it became pretty clear that inflammation and neuroimmunity were also a focal part, an important part of what the genetics are driving in terms of the disease etiology, as well as endocytosis and cholesterol metabolism. And here we are in 2022 with now 25 pathways, not 4 or 6 or 8. And each of these pathways has a number of genes in it, and each gene is in a number of pathways. So this makes it very difficult to discover therapeutic targets directly related to the disease. And this is a large part of the complexity. The ADSP developed a gene verification committee at the recommendation of NIH leadership in 2019. And they have been very carefully going through the literature to define what is and is not an actual Alzheimer's disease locus and what may or may not be an Alzheimer's disease gene. So this is now published on our NIAGAD's website. So you can look at all the entire list with definitions of what each of these things is. And again, to note that we are working towards endophenotypic definitions, loci and genes that are grouping to form endophenotypes. So we are kind of at a place now where we're at a bottleneck because we've done a fair amount of work in Caucasians, but almost nothing to mention in terms of ethnic diversity. Almost 90% of the reporting is in whites, as is seen in the data that are in NIGADs. And if you look at ApoE variants and how they modify risk, Europeans have a 3 to 4-fold amplification of risk associated with ApoE4 homozygous, whereas Africans have less risk and Asians have much more risk. So we have developed the ADSP follow-up study 2.0 to look carefully at factors such as this, and we're doing everything we possibly can. Everything that has been in the earlier parts of the ADSP is folded in here. And we're looking specifically at African, Hispanic, Latino, and Asian populations, as well as some Amerindian populations. This is a global effort. It includes whole genome sequencing, including long reads and GWAS. Our goal is 18,500 cases and 18,500 controls each in order to achieve statistical significance for rare variants for African, Hispanic, Asian, and Asian ancestry. So we think we'll have between 130 and 150,000 genomes by 2027 or 8. These will be case control, epidemiological, and family-based. And it's international. We have cohorts from India, Africa, Mexico, Central and South America, Korea, Asia, and the list continues to grow. For whole genome sequencing, our last release was a year and a half ago. Yes, a year and a half ago. 17,000 whole genomes, just about. And this week we have our next release that brings in 19,000 more whole genomes, bringing us up to 36,000 plus whole genomes available to the research community with growing numbers of ancestral diversity in the sequencing project. Just want to give a couple of examples of variation in AD risk, genetic risk that is associated with ancestry. The first one was pretty early on. It was 2015 when there was a frame shift identified by the Miami team, Peggy Perchek-Vance and her folks, and that was associated with Alzheimer's disease in African Americans. This was a very significant risk factor. It's actually more significant than ApoE4 for the African Americans. It's in an ABCA7 protein that's involved with APP processing in the endosomal compartment. And this frame shift was found in African Americans, but not at all ever that anybody knows in non-Hispanic white. So this is a very common ethnic specific variant that accounts for a good deal of the risk for African Americans. And just a point about diversity, we have basically two kinds of ancestry, two types of genomes to talk about. One is global ancestry. That's as a whole, seen as a whole, what the characteristics of a genome from particular regions of the world is viewed as. And then there's local ancestry, which occurs when people move into a very specific area. And then there's intermingling of the populations with the local population that's there. And we call that the local ancestry. If you look at the local ancestry for Europeans and ApoE4, those with African ancestry have a lower risk looking at local ancestry again with ApoE4, but Amerindians have a higher risk. So there's a number of things to look at with ethnic diversity. We have opportunities and objectives that we need to reach. The first thing is genetic risk factors in populations of diverse ancestry. We can now do genetic association and endophenotypes using our various clinical factors that we have available and comorbidities. cognition biomarkers, imaging, etc. Can develop population-appropriate polygenic risk scores, which we couldn't do before. We will be able to look at non-genetic risk factors, such as social determinants of health, including language, cultural, and educational differences, lifestyle, and other comorbidities. There are a number of gene exposure interactions that can be examined by comparing diverse populations. Biomarkers and diagnosis can be tailored, can be examined, can be specific for each of the ancestry populations. We can do multi-ancestry analysis and meta-analysis to increase statistical power for genetic discovery and compare across the ancestries. And we can assess protocols that are culturally appropriate and scientifically vigorous. With this, we can go to better clinical trial readiness for diverse cohorts. Just to give you a flavor of what we have coming into the cohort, a large multi-ethnic U19 has just been funded. This will be providing 4,000 Black Americans, 4,000 Hispanics, and 5,000 Africans. This is part of Hugh Hendry's cohort, the beginnings of the Nigerian group for Ibadan. But this is a continuation of that, in that there are now 9 countries and 10 sites in Africa that are using our protocols for data and sample collection. In the works is a global Latino sequencing project that is under peer review right now, and the numbers on Hispanics are growing. We have funded an ADSP foreign cohorts for Asia, and these include Korea, which has 2 cohorts, one led by Lindsay Farr and another led by Andy Sakin. These have similar phenotypic data, so we'll be able to cross-compare the 2 groups. We have the ASRI cohort, which is contains a few Aboriginal individuals in Polynesian groups, as well as some Caucasians, and a very interesting Asian group, the longitudinal aging study in Asia, La Ciudad, out of the University of Southern California. This one I just want to point out is different. We have found that the group cannot use the standard GWAS hits for comparison, and they have to have their own imputation panels because the genes loci that are coming out are very different from what we have in the European populations. There's also a large founder event in India. There is a lot of, particularly southern populations, have a great deal of inbreeding. So there are unique phenotypes that are emerging from this population. And of course, we have our own Lisan Wang, who's teamed up with a number of investigators across the United States to develop an Asian cohort for Alzheimer's disease. This will be about 5,000 participants, Chinese, Korean, and Vietnamese ancestry in the US and Canada. It's culturally appropriate with its recruitment and assessment, and the DNA for genetics and plasma are both being gathered. We have then this pipeline. We started in 2012. We added NIAGADs in 2012. Fortunately, the sequencing began in earnest in 2014. The Genome Center was launched in 2016. We began in earnest our diversity sequencing in 2017. This summer, we've added 4 initiatives. This past 2 years, we've added 4 initiatives. This summer, the diversity initiative here, the Phenotype Harmonization Consortium last year this time, Functional Genomics last year this time, or this spring more properly, and machine learning this last summer. So the linchpin of all of this is NIAGADS, the database. Any qualified investigator can obtain the ADSP data. Please have a look at this. There's an area where you can browse the genomic data and what the ADSP is developing there. If we now look at the next important thing to do, we've really got to align the phenotypic data carefully to understand the subtypes of the disease, the endophenotypes. So we have this large U24 phenotypic Data Harmonization Group, which is working very, very hard with continuous curation for the ADRD research community at large. This will be a legacy data set continuously curated through the years. The sources of data include epidemiology cohorts, our Alzheimer's disease research centers, and the new cohorts that are being generated that I just talked to you about. The types of data are case control, family-based, earlier onset Alzheimer's disease, autopsy, and neuroimaging initiative. Phenotypic and clinical data are cognitive imaging, longitudinal clinical data, neuropathological data, cardiovascular data, and biomarker data. And the consortium is working, the harmonization group is working in collaboration in waves of harmonizing phenotypic data and sending it on to NIAGADS. And as soon as NIAGADS is ready to do a data release, that group of harmonized phenotypic data is also released. We have onboarded the Functional Genomics Consortium with a goal to discover and characterize functional genomic and epigenomic elements to elucidate and validate the functional roles and to determine where they fit in the underpinning of the disease. These are very large scale studies, very high throughput, and a great team approach for this group. Generating stem cells for diverse ethnicities, looking at organoids through stem cells, nuclear sorting using RNA-seq and ATAC-seq efforts, CRISPR and CRISPR guide libraries, and a number of analytical tools, as well as developing AD mouse models. And our machine learning efforts under PAR 19269, looking at 10 areas of exploration that the NIA is interested in, and using all of the available data that I showed you before in this effort. So this effort is a global collaboration for the analysis of genetic and phenotypic data in more than 100,000 individuals. They're mostly generating research resources, sharing through GitHub. Algorithms are being generated, tools and resources to share with the research community, looking at diagnosis, progression, and conversion, as well as disease subtyping and stratification, along with drug target development, drug repurposing, and drug responsiveness. We have 8 teams, and I won't be able to tell you about what they're all doing, but here are some highlights. They have, among them, they have developed a deep learning method to classify Alzheimer's disease to predict MCI progression, looking at lipids as biomarkers to predict conversion of MCI to Alzheimer's disease, looking at expression levels associated with biomarkers, predicting gene regulatory mechanisms in various individual cell types and individual brain regions. They have a knowledge database. They have developed a comprehensive catalog of functional genomics and also have a post-mortem log of brain tissues. One of the more important efforts we have going is a collaboration with the Million Veterans Project. The MVP data, they have, as you know, 1 million genomes, 1 million veterans in their project. And not all of them obviously have Alzheimer's disease, but the problem is that their data are ICD-9, ICD-10, which doesn't really talk to what the NAC data are in terms of their electronic record keeping. So the VA is working together in teams to generate algorithms to convert the ICD-9, ICD-10 codes with reliability to what the ADSP is reading as a diagnosis for dementia and Alzheimer's disease. So here we are in our data production line. We're right here with our phenotypic harmonization, just the first release right now. They're actually a year ahead of where we thought they were going to be. Functional genomics having just launched. The first wave of AI/ML and second wave has been launched. And now we're moving into recruiting and retaining diversity cohorts and beginning to do the functional data for those cohorts as well. So here's the bottom line for everything. That's the ADSP. We have lots and lots of genetic and genomic data. We have lots and lots of phenotypic data and functional genomics in 10 different varieties. All of these data will be harmonized. We'll apply artificial intelligence and apply diversity to this. And what we will come up with was an AD phenotype that is defined by a genetic profile. What we want to do, and we know we're going to have many of them, but we want to define the disease by the subtypes of the disease. And the reason that we want to do that is because we've had more than 400 clinical trials so far, and most of them have not shown very much promise. And the reason, probably at least in part, is that we're studying Alzheimer's as a whole. We don't want to do that. We want in our clinical trials specific targeted groups of individuals with specific genetic profiles. This will then help us to select individuals that are fully characterized down the line for endophenotypes in clinical trials. So where are we now? We know with certainty Alzheimer's is a genetic disease. It is inherited. The Alzheimer's disease is a spectrum of comprised of subsets or endophenotypes that are likely to be genetically driven. There's early onset and late onset, no more. We have earlier onset and later onset as a continuum. Most of the variants are rare or very rare variants, and they're almost all in the non-coding region of the genome. These are pointing to major pathways such as inflammation, lipid metabolism, endocytosis, and amyloid deposition, but also diversity is going to be extremely important to this. So we are putting our shoulder to the wheel with immediate objectives of the robust expansion of multi-ethnic cohorts, development of new analytical approaches, and broad sharing with the research community. So we now have over 70 loci identified by the AD community in the last decade. We have causal pathways and mechanisms linking AD-associated alleles to disease manifestations being explored. We have diverse ancestry populations growing. We have AI/ML methods to approach gene discovery and fine mapping. And we have genetic findings that have been nominated multiple targets that are moving towards clinical trials. Thank you for your attention, and I look forward to your questions.